Should have kept it a surprise.
Why should I kept it a surprise? Take it home and like, hey, look what I found.
You know what they say? Ask for forgiveness, not permission.
Permission? Yeah, maybe I should have done that, huh? Anyways, so moving on with today's material. So as far as your test, honestly, I'm not sure when I'm going to get it graded. I'm going to try, but I make no promises because your guys' test was not the only test that I had yesterday. My physiology and my conservation biology also had tests yesterday. So there's a lot of grading for me to do, and I'm a little behind on lessons as well. So don't be surprised if I don't get these graded as quickly as I grade some of the stuff. I will do my best, but I make no promises as to the speed of my grading this time. Usually I grade tests pretty quickly, but this time I am not sure. But we will see what happens. But we are starting mitosis and meiosis today. We are going to have a focus on mitosis first, and then once we learn mitosis, we will do meiosis next. So this is how cells reproduce, and looking at specifically, we've already learned replication, and replication is how the DNA splits, and then we're going to look at once the DNA splits, what happens to the actual cell and how the cell is going to split apart. So this little video clip shows the process of, I wonder if this one has sound. It didn't have sound last time. So then I think we skipped to this one, which did have sound.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatin toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
So the cell cycle is from one division to the next division. So when a cell divides to when the cell divides. Because remember, cells don't just miraculously appear. Each cell comes from pre-existing cell. So it's a series of events from when the cell divides to when the cell is going to divide again. That is the cell life cycle. So within that cell life cycle, a lot of things are going to take place step by step. And again, it is the replication is just a small part of that, which we learned about last time. eukaryotic cells undergo mitosis and or meiosis. So meiosis is for reproductive cells only. Not all eukaryotic cells are going to undergo meiosis, and we'll talk about why in a little bit. But prokaryotic cells undergo something that's a little bit different. They don't undergo mitosis, they undergo something called fission. And this is a little bit of a separate process, so a little bit different. So they had the TV up yesterday. Yeah. Apparently it works. Not broken. I guess fourth time's a charm. But the problem is it doesn't talk to the computer. So, it's up, but doesn't talk to the computer. That's okay, baby steps. They also took the projector down.
Oh boy.
Yeah, so no hanging projector.
Oh.
And I have a new tile. That's kind of fun.
That title's different.
What?
So it's a different title.
It is.
Yeah.
But I'm still missing a tile here. It is a different tile. So now somebody needs to paint that one for me. Otherwise it's just weird. Goodness, I'm tired. Now this is in Google Classroom, but it's basically the difference between mitosis and meiosis. Mitosis is the basis of growth and cell replacement and tissue repair. So for an organism to grow, it needs to go through mitosis. If an organism is injured, then, like say you get a paper cut, and there is now a gap in your skin needs to fill that gap. And the way it fills that gap is making new cells to put in that space. So dividing the cells nearby, and those dividing cells are going to fill in that hole. Mitosis. It's also the basis of asexual reproduction. So if an organism is a single-celled creature and it wants to reproduce, then it just divides in half and boom, it's made a copy of itself and it's reproduced. Now meiosis is a little bit different. Meiosis is the basis of sexual reproduction. Meiosis only takes place in reproductive cells. So we're talking egg, sperm, pollen. Egg, sperm, pollen, reproductive cells, that's where meiosis takes place. Mitosis is genetically the same, identical. Meiosis, different. We want different for meiosis. Mitosis the same, meiosis different.
Robert Remak is not one of the best known names in the history of science, but for me, he's a genuine hero for our story. Remak was Jewish, so attaining the professorship he deserved was always going to be an uphill struggle. He was forced to do his research in a dingy attic apartment. Despite these obstacles, Remak set out to discover how new cells originated. He realised his best hope was to look in a place where he'd be guaranteed to see lots of cells forming. Professor Reddy's has helped us recreate Remac's experiment from the 1840s.
The chick embryo was the animal model of choice for embryology at the time, because eggs are very easy to get, they're very inexpensive, and also the embryo is accessible. That means you can now take some scissors and cut a hole here on top. And there it is. The embryo floats on top.
Okay.
And we're going to cut a blood vessel now and collect a little bit of blood. So I have a glass pipette here, and I'm going to suck a drop of blood. And here you can actually see that there is red fluid in the pipette. It contains blood cells, red blood cells. Putting them on a glass here. And to see them better, we put a cover slip on top so that we can look at it under the microscope. Here you go. Now let's have a look. Take the microscope here, which is of the type that Remarque may have used. This is a really old one. I have to adjust the light source with the mirror here.
Not the type of scope that you're used to using.
Well, the scopes we're using are a bit more modern now. You can actually look with both eyes. I will have to close one eye and look through here. And what you can see actually is many blood cells. They're still floating around a little bit because the fluid is moving. They're round, most of them. But occasionally I see a cell in division, which is not round, but which has I don't know the technical terms in English. There is some invagination of the cell membrane.
That's the same word, invagination.
Yeah.
So it begins to split into two like that.
Right.
So it invaginates at both sides.
Yes. That means cells separating from each other in this stage, which takes a long time. So you cannot see the whole process because it takes several hours. But you see snapshots of this process. Have a look.
So it's not actually obvious. He must have been very persistent to see the individual stages from one cell as it begins to sort of fold in on itself and then divides into two cells. He must have done this experiment a lot of times.
He must have used hundreds of eggs actually. And what's amazing is that what caught his attention were the few cells, the very few cells that were in division. They may have easily been missed by some other researchers who may have thought that these few cells in division are some artifacts under the microscope. But he focused them then and systematically studied them. And these stages in division are actually those that he depicted in his publication.
You look at the surroundings that Remarque was working in, which is a sort of dusty old attic. Well, it's amazing that he came up with the results that he did.
No, that shows you that sometimes the instruments themselves do not advance science, but the thinking of people and having new ideas is more important than good equipment sometimes.
And so Remarque is a true pioneer.
I would say he's one of the heroes in science, in cell biology, because he really persisted on this idea and supported it by very well-founded observations.
So really kind of cool that, I mean, I don't know if you've ever tried to look at one of those very basic microscopes. They're pretty tough. I mean, it's sometimes hard to find things under our cool microscopes, but if you try and look at something under those really like basic microscopes, it's even harder. So kind of neat that he was able to do so. Anyways, this chart is in Google Classroom. It looks at the mitosis, meiosis, and then prokaryotic fission, and kind of gives the description of what's going on in each one. So it's just a good comparison and in Google Classroom for you. Did I post the student notes? Maybe. I can't remember. I think I did. So anyway, chromosomes and dividing cells. We already mentioned that people have 46, 23 from mom, 23 from dad. We got curious fifth hour and looked up what had the most and what had the least. Apparently there is a fern that has over 1,000 chromosomes. It's like 1,400 or something, like 720 from one parent and 720 from the other parent. And then there is an Australian ant that has 1 chromosome. The male has 1 chromosome, and the female has 2. So that was rather interesting as well. Chromosomes differ in length and shape and carry different portions of cells' hereditary information. And we already mentioned that a little bit last unit with that material, and how the chromosomes they vary, but they have very similar information. Like the one you got from mom for hair color, and then there's one from dad for hair color, and so on. So basically, when a cell divides, it's going to split in half, and each cell will have replicated its DNA. So it's got, for a while, instead of 46, it has 92 chromosomes. And then those 92 are going to divide out to 46 again, and that's going to separate out into 2 cells, and then that 1 cell is going to split into 2. And it's going to make sure that each of the new cells has enough cytoplasm, has enough smooth ER, has enough Golgi bodies, and all of that other stuff. So the first step is called interphase. Interphase is what the cell spends most of its life cycle in. Interphase is the very beginning, and it is when the cell is doing its cell stuff. It's the cell being a cell. It is like, if it's a skin cell, then it's being a skin cell. If it is a stomach cell, it's being a stomach cell. If it is a kidney cell, then it's being a kidney. It's just being a cell. That's what it is doing. So interphase is the longest because that's when it's just doing its stuff. The other phases of mitosis, the prophase, metaphase, anaphase, telophase, which you've hopefully at least sounds familiar, that is a very fast process. So of the cell life, the actual division process takes like 45 minutes to an hour or so. So you have to be very quick to catch it during that transition phase. We did the cheek cells, right? Looked at the cheek cells? Yeah. You probably didn't see anything in mitosis, because to catch the cells actually going through a division process, probably the only way you caught one in mitosis is if you had, like if you bit in your cheek and you had a sore in your cheek. And then possibly something was going through mitosis because it was filling in that sore.
I have a cold sore right now. You know what I'm doing?
It's possible then, but it's more likely like if you had bitten it and you had an injury. Now in interphase, it can be subdivided into 3 stages, G1, S, and G2. G1 is just the normal stage. It's metabolic activities, the cells just do its thing. S is when the DNA replicates itself. S stands for synthesis. So this will be a question on your next test. When does the DNA go through replication? And the answer is the S phase of interphase. So when does DNA go through replication?
S phase.
S phase of interphase. That will be a question on your next test. DNA goes through replication during the S phase of interphase. G2, by the time we get to G2, the DNA is already replicated. It's hanging out in the nucleus as 2 parts because we made a copy. Then in G2, the cell is kind of checking itself. Do I have enough cytoplasm Do I have enough proteins? Do I have enough Golgi bodies? Do I have enough smooth ER? Can we actually go through with the cell division process? Is there enough stuff to go through with this? That is kind of what the cell is thinking to itself. Do we got enough? And if not, then in G2 the cell is going to make more. So it's still doing some like being a cell kind of stuff, but in G2 it's just double checking. It's like, okay, we're big enough now. We've gone farther than we need to. We got you ready to divide. Let's do this. Do we have everything we need to do this? So mitosis is the process of nuclear division that maintains the chromosome number identical. That is the big deal about mitosis. Everything needs to be the same. Mitosis is the same. Now when we get to meiosis, we're going to see it's different. That's the point of meiosis. Meiosis, we want different. We don't want every single egg and sperm to be exactly the same. That is not the point of meiosis. Meiosis is different. We want different. Mitosis the same, meiosis different. So mitosis and cytoplasmic division are the basis of developmental processes, increasing body size. That's how we get bigger. We don't get bigger because our cells are bigger. We get bigger because we have more cells. Replacing of damaged or dead cells, and also, again, asexual reproduction.
Poppy Spry, please report to the office. Poppy Spry, the office.
I really like this picture of the visual of how mitosis works. I think it's really good at explaining it and putting it in a way that's easy to understand. So if we start here, imagine this is where the cell life begins. The cell has just divided. And so we start from one division. So if a cell is just divided, this is where the life of the cell starts. So this whole section here, this purple, is all interphase. And you can see the interphase takes up a huge portion of the cell life cycle. So most of the cell life cycle is interphase. We've got G1, S, and G2. So G1 is just the cell growing, just, doing its thing, just being a cell. And then we have replication, which is the S phase. So the DNA is replicating itself just like we learned about last unit. Then we've got G2, which is now the DNA is replicated and it's like, do we have enough of everything we need? Is the cell at a point where we can actually make this happen? If the cell is like, yeah, we're good to go, then it's going to go into mitosis, which is the cell division phase, okay? So this little chunk here is the cell division phase. And again, this happens really, really quickly. So about 45 minutes to an hour. In the entire life cycle of the cell, this is a very fast process. We've got prophase, metaphase, anaphase, telophase, and then we have cytokinesis. The other thing I really like is that it shows that cytokinesis and mitosis overlap. Cytokinesis is the division of the cytoplasm, which we'll get into later. It's a separate process. It's not the same. Mitosis and cytokinesis are two different things, but they do overlap, and I like how this picture shows that here. And they also take place at about the same time that telophase is taking place. So you have all these things going on at about the same time right here. Mitosis does consist of prophase, metaphase, anaphase, telophase. Mitosis is not cytokinesis. Cytokinesis is different, but it happens at the same time as telophase. So I drew up here on the board IPMAT-C. I for interphase, and then prophase, metaphase, anaphase, telophase, cytokinesis. IPMAT-C. That's how I always remember it. So the cell cycle, the red, goes from interphase to cytokinesis. The whole cell cycle is interphase to cytokinesis. Then PMAT is mitosis, because mitosis is just the division of the DNA and the nucleus. That's it. DNA and nucleus is PMAT. Cell cycle is from I to C. PMAT is mitosis.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into 3 discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the 2 groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of 2 genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase. And in these phases, duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of 2 genetically identical daughter cells.
So 2 daughter cells are the result of mitosis. You start off with 1 parent cell, and by the time you're done, you get 2 daughter cells. Again, you have the same number, you have the same kind, everything is the same. That is the point of mitosis. Everything is genetically the same. You want them to be the same. They are also something called diploid, di for 2, or it can be written as 2N, containing the 2 pairs of chromosomes, 1 from mom, 1 from dad. So a full total of 46 for humans, or 1,440 for that weird fern. So a full set of chromosomes means 23 from each parent for humans. Now, we will learn about haploid for meiosis, in a haploid cell, the haploid are gametes, because you don't want an egg and a sperm to fuse. If an egg and a sperm are both 46, and they fuse together, like an egg that's 46 and a sperm that's 46, when they fuse, they're going to be 92. And then later on, when that individual has a 92 and a 92, what is it, 184? And then it's just going to keep increasing and increasing and increasing. So that's what happens if you don't make the numbers smaller. So in meiosis, you make a smaller egg or sperm. The egg has 23, the sperm has 23. So what? That way when the egg and the sperm fuse, 23 plus 23 equals 46. You are keeping that same number. So that's one of the main differences between mitosis and meiosis. Now we also have the homologous pairs. We talked about homologous. These are the ones that have the same length, the same color, the same shape. Everything is the same except you got one from mom, you got one from dad. You got one for being how tall you are from mom, you got one for how tall you are from dad. My children have this weird talent of being able, like their toes are double jointed or something. I don't know, it's really weird. But they can curl their toes under in a very weird fashion.
Sounds really gross.
It's very gross. Like, it's not even like the toes. It's like the next joint down, they curl their toes. And their dad can do that. And he's always been able to do that. And all my children can do that. I can't do that. My toes don't work that way. But that is an example of a chromosome they got from him somehow. And so they got that from him. That is a homologous chromosome. My homologous chromosome does not do that. Weird traits. Genetics is so fun. I can't wait to that. That's our next unit, genetics. It's going to be a fun unit. I really like genetics. Alright, so the sister chromatids, remember that X we looked at before, those are sister chromatids when they are joined together in that X and they are genetically identical to each other. One of those is exactly the same as the other one. They're just condensed. And I think I explained why it's important to condense them because, no, I don't remember if I explained it. But basically, like, you have to condense them because it's easier for them to move around. If they are left long and stringy, not easy to move around. If they are condensed, much easier to move them around. Long and stringy, not so good to move from place to place. Condensing, good to move from place to place. Goodness, I'm tired. So during mitosis, the sister chromatids are pulled apart. So they get separated. They were in an X, and they are no longer in an X, and they get pulled apart. You can see how they are homologous, like this one looks the same as this one looks the same as this one, and so on. They get pulled apart during mitosis. And we'll get into more details like that. Did you guys learn this in middle school? Yeah, okay, good. So not totally lost. They get pulled apart during mitosis. Then which phase is this? If you learned it in middle school, we didn't talk about it yet.
Oh, I don't remember. It was 8th grade.
8th grade. It's been a while.
Yes, this was Mr. Mackey.
I'm
Pretty sure.
bored.
Interface.
There you go. Will you remember it by the time I'm done with you? Pound it into your heads. This is in Google Classroom. It's just kind of a brief description, but it's showing that you start off with one cell. Here's what you start with. Makes a copy of its DNA. It's replicated. You got a sister chromatid here. And this is what you end up with. It looks exactly the same. Same, same, same. These are the same. This is the same. This is the same. It's all the same. This one's not because it's duplicated. It's replicated itself. Same. It's all the same. That's the point of mitosis. Everything's the same. It's all the same.
What is it?
It's the same.
Pardon the interruption. Students who are juniors and seniors that are interested in education or teaching after you graduate, please come to my office. That's juniors or seniors interested in education or teaching after you graduate, please come to my office. Also, this is the last day to sign up for ACT this spring. So juniors and seniors interested in the ACT this spring, today is the last day to sign up.
When you guys have the opportunity to take it next year, take it many times. As many times as they offer that test, take it. I don't care how annoying that test is. If it's free, take it. Took it 6 times, the ACT. Okay, I needed a certain score and I finally got my score that got me free scholarships. Paid for all my schooling. Alright, anyways. Whether or not a cell divides is determined by mechanisms of gene expression control. So there are things in the cell that say, okay, we need to divide. Or, okay, yeah, let's not do it right now. It keeps the vast majority of cells in G1. G1 is the, okay, I'm just being a cell. And that's where most of the cells stay. I mean, that's where they're supposed to stay. So they're supposed to stay in G1 unless something goes wrong and then it gets out of G1 and it gets out of control. Unless the cell is supposed to divide. Anyways, checkpoint genes are what these are called. The checkpoint genes, they will say, okay, you may go on to mitosis, or nope, you need to stop. And usually, we already mentioned this, they kill the cell if the cell is dividing and it's not supposed to. So basically, the cell does not need to divide, or if it is not ready to divide, it is not allowed to go on to the next step and divide. It only divides if it needs to. It only divides if it's ready to. If there's a mistake, it doesn't divide. Checkpoint genes prevent mistakes from being allowed to continue. I've got to stop yawning. I know I've got to go to bed earlier.
What time did you go to bed?
About 11. But I wake up at 5:15. And I went on like a 2-mile hike, probably like 2.5 miles. Actually. So, multicellular organisms, the mitosis and cytoplasmic division are a basis of body formation and increases of size. That's why little people divide faster. I mean, children. I'm referring to children. Children divide faster. They go through mitosis a lot. If we looked at my kids' cells versus my cells, they would be growing really fast. Mitosis is the part of asexual reproduction, and that's how little tiny organisms make offspring. But we already mentioned both of those things, which is why they're not in red. You don't have to write them down. And we're done. Look at that. You guys finished faster than the other class. They worked right up into the bell. You guys are just so much faster. So, all right. I them the other assignment, so I'm not gonna give you the other assignment yet, just to keep you guys at the same pace. So you guys are just lucky that the other class is slow. So I just like to keep you at the same pace as them. So I'm not gonna give you the assignment. What?
That'll hold us back.
It doesn't hold you back. I mean, do you want the other assignment?
We're good.
I figured that was the case. You can have it if you want it.
We'll let them catch up.
You'll just, let them catch up.
Just that nice.
We'll see if it's an assignment I need to print off or if it's in Google Classroom. I have a note in my notes. See, my note says print off. I need to print it off or not. I don't have it printed off. Closest review. Yeah, I should probably print this one off. Fine. How many people do I have? 13 in this class and 4 in here. That's what, 17?
Yes.
I
18.
guess I'll print it off. 18. I mean, it wouldn't hurt to make 18. That way I can have it.
More money for you. What? She's paid more based on how much for her class. Well, yeah, but there's 17. There's 18. She paid more.
That's not why I want people to take my class. I want people to take my class so I can help you. Jeez. It's not all about money.
Until you don't have enough money.
Well, I just went how many years was it without my husband having a decent job? At no fault of his own. I mean,
May I go get some more ice upstairs?
yes,
Can I get some more water?
yes.
Thank you.
There's just a
And on both
Nah.
sides. Okay. Good. Printing. There we go. Now we'll be ready for tomorrow. Kylie, are you okay? You're very quiet today.
Yeah, I'm tired.
Say maybe it's just because Sam is in here and you don't have your talking buddy.
Might be a little poor, but
You just seem quieter than usual.
yeah, I'm just really tired.